Estimation of time since death, Part 2. I'll again begin with the quote by Dr. Gyan Fernando. A good pathologist holds knife just like a violinist holds a bow. The learning outcomes of this particular module will be, as you have studied for estimating time since death, there are immediate signs, early signs and late signs or the changes. Immediate and part of the early changes we have already discussed in the previous module. In this particular module, we will be discussing amongst the early changes after death, the rigor mortis, the mechanism of production of rigor mortis and governing factors, the late changes after death, that is decomposition including putrefaction and autolysis, and the altered process of decomposition, that is Adipocere and mummification. Coming on to the rigor mortis. The rigor mortis or death stiffening or cadaveric rigidity is a state of stiffening of muscles, sometimes also with slight shortening of the muscle fibers. During life, the muscles are in a slightly contracted stage. After passing through the primary relaxation phase or primary flaccidity soon after death, during which the muscles are totally relaxed, the muscles gradually get stiffened over a period of time, which is known as rigor mortis. The rigor mortis is a physicochemical reaction which affects all the muscles of the body, both skeletal and smooth. A voluntary muscle is made up of bundle of long fibers. Each muscle fiber in the bundle consists of densely packed myofibrils, which are the contractile element. And these myofibrils are made up of two protein filaments, actin and myosin. During lifetime, the separation of actin and myosin is possible for the extensibility of the muscles. The contraction of the actin and myosin filaments are dependent on the energy provided by ATP, that is adenosine triphosphate. And during life, a constant supply of ATP is being maintained. This failure of resynthesis of ATP after death results in the muscle becoming hard and rigid. Some amount of ATP still remains available after death till the glycogen reserves lasts. Once the glycogen reserve is depleted, there is no more ATP available and the contractile elements are converted into dehydrated, stiff, gel-like mass, resulting in the muscles themselves becoming hard and rigid. Then there is a definite pattern of appearance of rigor mortis in the body. So let's understand what is the order of appearance. All the muscles of the body, both voluntary and involuntary, are affected. The Niston's rule states that it does not start simultaneously in all muscles. It first appears in the involuntary muscles. The myocardium becomes rigid in about an hour. It becomes noticeable in the eyelids, then the muscles of the face, jaw and neck. Then it goes downwards to the upper limbs, thorax, abdomen and the lower limbs, and lastly the terminal parts, that is fingers and toes. In the individual limbs, it usually progresses from above downwards. It disappears in the same order in which it has appeared. With the passing away of rigor, the muscles once become softened and this is called as secondary flaccidity. If you see the development of rigor mortis, it is generally considered that by first 12 hours after death, the rigor affects the whole body. Then it is retained for another 12 hours and passes off in the next 12 hours with the onset of putrefaction. So this 12 hours is called as the rule of 12, which is also known as march of rigor. However, in temperate climates, it begins 3 to 6 hours and takes further 2 to 3 hours to develop. The conditions altering onset and duration, there are which alter that appearance of the rigor mortis. The rigor mortis does not occur in a fetus of less than 7 months. In children and old people, it is feeble and rapid. Onset is early and duration is short in cases where death is from diseases causing great exhaustion and wasting, for example, cholera, typhoid, tuberculosis. Onset is delayed in deaths due to asphyxia, severe hemorrhage, apoplexy, pneumonia, nervous diseases, all these conditions. In death due to strychnine or hydrocyanic acid poisoning, it starts early and persists longer. It may disappear very rapidly in case of bacterial infection, gas gangrene. In case of healthy muscles, duration is long and onset is slow. If there is fatigue or exhaustion, onset is rapid with short duration. In cold weather, onset is slow and duration is long. If the body is in extremely hot condition, the rigor mortis may disappear in 12 hours after death. Medico-legal importance of the rigor mortis is, it is the sign of death. It helps in estimating time since death to some extent. It indicates the position of the body at the time of death to some extent also. Then mimicking with the rigor mortis, there is a condition called cadaveric spasm or instantaneous rigor. The cadaveric spasm is the rare state where instead of primary flaccidity after death, the muscle usually belonging to the small group go into sudden state of stiffening. The exact mechanism is not known, but possibly neurogenic and not the same chemical process as rigor mortis. The muscles that are contracted during life become stiff and rigid immediately after death without passing into a stage of primary relaxation. Hence, the change preserves the exact attitude of the person at the time of death. Now, these predisposing factors for cadaveric spasm are, they occur especially in cases of intense physical or emotional activity such as excitement, fear, severe pain, exhaustion, injury to the nervous system, firearm wound of the head, the drowning, the convulsant poisons such as strychnine, all these. This is usually limited to a single group of muscles and most commonly involves the hand, the smaller muscles. For example, as we see in drowning cases, the grass or weeds may be clutched in the hands, or in suicide by shooting, the weapon may be clasped in one hand. No other condition simulates cadaveric spasm and very great force is required to overcome this stiffness. The medico-legal importance of the cadaveric spasm is that the weapon of offense in cases of suicide can be seen grasped in the hand. If the deceased died due to assault, then some part of the clothing of the assailant like button or hair may also be seen firmly grasped in the hand. In drowning, grass or weeds may be clutched in the hands, which indicates that the person was alive on entering the water. The cadaveric spasm helps in indicating the mode of death, that is, whether suicide or homicide or accidental. Let's understand the difference between the rigor mortis and the cadaveric spasm. If you see the mechanism, in case of rigor mortis, the mechanism is well known due to depletion of the ATPs, whereas cadaveric spasm mechanism is still not clearly known. Predisposing factors for rigor mortis is nil, none. It's a usual process after death. Whereas in cadaveric spasm, the predisposing factors are sudden death, excitement, fear, exhaustion. Usually, the time of onset in case of rigor mortis is 1 to 2 hours after death, whereas the cadaveric spasm, it occurs instantaneously. In rigor mortis, both voluntary and involuntary group of muscles is involved, whereas in cadaveric spasm, it is usually restricted to the single group of voluntary muscles. The muscle stiffening is not much marked in case of rigor mortis, whereas it is very well marked in case of cadaveric spasm, and the great force is required to overcome it. Then, in case of rigor mortis, there occurs molecular death, whereas in cadaveric spasm, the molecular death does not occur. If we do the electrical stimulus, the muscles do not respond in case of rigor mortis, whereas in case of cadaveric spasm, the muscles will respond to the electric stimulus. If we see the muscular reaction, in case of rigor mortis, it will be acidic in nature, whereas in case of cadaveric spasm, it will be alkaline in nature. Coming on to the medico-legal importance of rigor mortis, it basically indicates the time since death, whereas the cadaveric spasm indicates the mode of death, that is whether suicide, homicide or accidental. Then there are certain other conditions which resemble rigor mortis, like heat stiffening, cold stiffening, gas stiffening, all these. So let's see what is heat stiffening. When the body is exposed to temperatures above 65 degrees centigrade, heat stiffening is produced, which is much more marked than the rigor mortis. Heat causes stiffening of the muscles because the tissue proteins are denatured and coagulated as in the cooking. The muscles are contracted, desiccated or even carbonized. A zone of pinkish brown cooked meat is seen under this, overlying the normal red muscle. The stiffening remains until the muscles and ligaments soften from decomposition. Then the cold stiffening. The tissue becomes frozen and stiff when the body is exposed to freezing temperatures before acid metabolites appear in the muscle. This is because of freezing of the body fluids and solidification of the subcutaneous fat, thereby simulating rigor. The body is extremely cold and markedly rigid, and if joints are forcibly flexed, ice breaks in the synovial fluid with a sudden snapping sound. The stiffness disappears if the body is kept in warm atmosphere. Then there is gas stiffening. The gases evolve during the putrefactive process. They also stiffen the body, and usually this poses no problem in distinguishing from the true rigor. Then after finishing with the early changes after death, now let's understand what are the late changes after death. That is primarily the decomposition, which involves two processes, putrefaction and autolysis. The putrefaction is due to fermentation by bacteria. After death, the bacterial flora of the GIT is spread throughout the body, thereby producing putrefaction. At cellular level, putrefaction begins immediately after death, but changes are not visible grossly. The gross changes are visible externally after the rigor mortis has passed off. So in summer, however, the changes may appear early. The bacterial enzymes are derived mostly from the anaerobic organisms from the intestines, mainly being Clostridium welchii. Others are Bacillus aerogenes, Bacillus capsulatus, Bacillus coli, Bacillus proteus, Staphylococcus and Streptococcus. The entry point of the bacteria is GIT, respiratory tract, genital tract and open wounds. GIT, of course, is the most common. The spread of the bacteria is through the bloodstream. The other sources of enzyme are fungal, like Penicillium, Aspergillus, and the larvae of insects also. The enzyme lecithinase, which is produced by Clostridium welchii, plays a very important role. This lecithinase hydrolyzes the lecithin of cell walls and RBCs, causing the post-mortem hemolysis. There is also an extensive and progressive breakdown of soft tissues, including proteins, carbohydrates, and fats. The factors which favor the growth of the bacteria are decrease in the oxygen concentration in the tissues, increase in the hydrogen ion concentration and proteins, carbohydrates and fats of the body, which act as excellent culture media. Then the second phenomena under decomposition is autolysis. The autolysis is the breakdown of cells and organs through an aseptic chemical process caused by intracellular enzymes. Soon after death, cell membranes break down with release of cytoplasm containing enzymes, that is glycolytic, proteolytic, lipolytic. These enzymes, they auto-digest the tissues even in the absence of bacteria. The process is accelerated by heat and slowed by cold. The organs rich in enzymes will undergo autolysis faster than the organs with lesser amount of enzymes. For example, pancreas autolysis earlier. The autolysis causes maceration of the dead fetus in utero. Then changes occurring during putrefaction. What are those? The first change, the greenish discoloration in the right iliac fossa, is the earliest external manifestation of the onset of putrefaction, which usually occurs about 12 to 24 hours after death in our Indian circumstances, environment. This is the area where the cecum with contents rich in fluid and bacteria lies. So that's why discoloration, greenish discoloration occurs at the right iliac fossa. The hydrogen sulfide produced by the bacteria basically acts on the hemoglobin, which has now become extracellular due to hemolysis, and ultimately self-hemoglobin is produced. And this self-hemoglobin imparts the greenish blue color. And with passage of time, this greenish blue color spreads to the abdomen subsequently and also makes its appearance in the chest, face, and later on in the limbs as well. The coalescence of these colored areas result in the entire body becoming greenish or ultimately greenish black. Later, in about 36 to 48 hours, a network of veins become visible in the form of a characteristic mosaic-like pattern which is called as marbling over various parts of the body like root of the neck, shoulder, where the veins are lying superficially, so they are easily visible. This results from staining of the vessel wall due to diffusion of hemoglobin. By the time the color changes become visible externally, evolution of gases would have started internally, which possess very unpleasant smell as well. The gases of putrefaction are hydrogen sulfide, ammonia, carbon dioxide, carbon monoxide, and methane. These gases by themselves are of not much medico-legal significance, but the pressure effects produced by them bring forth the gross changes in the body. The evolution of the gases result in the ballooning effect as well as dispersion of the post-mortem lividity. So what is ballooning effect? The gases basically easily collect in the hollow viscera, resulting in their voluminous expansion. The stomach and the intestine, which balloon out, increase the intra-abdominal pressure, resulting in the distension of the abdomen and pushing the diaphragm upwards. The lungs are squeezed by this pressure and the froth oozes out from the mouth and nostrils. The gases not only accumulate in the hollow viscera but also penetrate into every tissue and organ. The soft tissue of the body like breast, scrotum, periorbital tissues, etc., they increase in their size enormously. The eyeballs protrude out of the sockets. The cheeks, the lips, they puff out. The tongue protrudes through the mouth and on exposure to air becomes dark in color. The facial features completely altered, and at this stage identification by external feature becomes difficult. The percolation of the putrefactive gases in the undersurface of the skin results in the epidermal layer ballooning out, which resembles the blisters as happens in scalding. But in contrast to the protein-rich fluid present in the blister which are caused by scalding, putrefactive blisters contain only air. Epidermal layers from the hands and feet slip off like glove and stocking. And this slipped off skin is important to preserve as it can yield full set of fingerprints that may help in establishing identity from the tip of fingers. This increased pressure inside the abdomen may result in the expulsion of fetus in the case of the deceased being female who was pregnant. The uterus and rectum may protrude out. The evolution of gases commences as early as 18 hours and it takes 48 to 72 hours to reach the full-blown picture. Then the dispersion of postmortem lividity. This evolution of gases moves the blood in the blood vessels. If the blood is clotted, it liquefies and hemolysis takes place. The movement of the blood results in dispersion of postmortem lividity in any direction. Therefore, assessment of the position of the body at the time of death based on the lividity at this stage becomes difficult. Then there occurs collicutive putrefaction or collicutive liquefaction. In 3 to 5 days after death, the nails and hair becomes loose and can be pulled off with ease. The teeth become loose and can be pulled out from the socket. The abdomen, thorax burst open and skull sutures get loosened, through which the liquefied brain matter oozes out. With further progression from 5 to 10 days, the tissues and organs are converted into soft, thick, blackish mass, totally losing their architecture. However, even at this stage, organs such as the prostatic and non-gravid uterus can be identified as they resist putrefaction, thereby helping in identifying sex of the individual. This liquefied, unrecognizable, pultaceous mass gradually falls off from the bony attachments, leaving the skeleton exposed. If a dead body is lying in the open, it may take 1 to 3 months for complete skeletonization. If the body is buried, the skeletonization may take place in 2 to 6 months. In case of body encased in coffin, the same may take more time. Sometimes environmental condition arrest the progression of putrefaction and may deviate to either adipocere formation or mummification. So now let's understand what is adipocere. Adipocere or saponification or grave wax. The adipocere is a modified form of putrefaction where the fatty tissues of the body change into a substance known as adipocere. This change is mainly due to the hydrolysis and hydrogenation of the body fats such as olein into higher fatty acids, which combine with calcium and ammonium ions to form the insoluble soaps, which being acidic inhibits the putrefactive bacteria. Ultimately, the whole fat is converted into palmitic, oleic, stearic and hydroxysteric acid together with some glycerol, and a mixture of these substances forms the adipocere. At the time of death, the body fat contains about 0.5% of fatty acids, but in adipocere they rise to 20% within a month and over 70% within 3 months. The process starts under the influence of intrinsic lipases and is continued by the bacterial enzymes of the Clostridia group, mainly Clostridium perfringens, as the bacteria produces lecithinase, which facilitates the hydrolysis and hydrogenation. The prerequisites of adipocerous changes of the body fat are the damp and warm environment, humid climate, still air, abundant body fat, warm temperature, and bacteria producing fat splitting enzymes like Clostridium falci. Thus, a body lying on the damp soil in a shady place with optimum temperature and no free air current blowing over it undergoes adipocerous change. Adipocere has the appearance of rancid butter and gives off a sweetish but disagreeable smell. The transformation of the body fats usually seen over fatty areas such as cheeks, breasts, buttocks or limbs. Rarely whole body may be involved. It takes generally 3 weeks for adipocere to fully develop. However, complete conversion in an adult limb requires at least 3 to 6 months. The same may persist for a year or even a decade. As the progression of the putrefaction is arrested, the facial features are recognizable, which helps in the identification of the deceased. Similarly, wounds are also preserved without much alteration and hence the wound can be studied properly. The proximate time since death calculation becomes impossible in this situation. The medico-legal importance of the adipocere formation lies in the fact since as the features are well preserved, the deceased can be identified even after much time. Time since death cannot be calculated for the same reason exactly. The cause of death can be determined because the injuries are well preserved if they are there. Then the second altered phenomena is the mummification. It is the modified form of putrefaction where the dehydration or drying and shriveling of the cadaver occur from the evaporation of water, but the natural appearances of the body and general facial features are preserved. It begins in the exposed parts of the body like face, hands and feet and then extends to entire body including the internal organ. The skin may be shrunken, contracted, dry, brittle, leathery and rusty brown to black in color. Is stretched tightly across the anatomical prominences such as cheekbones, chin, costal margins and hips. Adheres closely to bones and often covered with the fungal growth. As the skin contracts, some of the fat cells in the subcutaneous tissue are broken and the liquid aisle is forced into the dermis, which becomes translucent. The entire body loses weight up to 60 to 70%, becomes thin, stiff and brittle. In mummified bodies, the joints are often seen in a flexed and abducted position, mainly due to shrinkage of muscles and tendons. Sometimes this phenomena may be partial also. If a mummified body is not protected, it will break into fragments and eventually will become powdery. But if preserved, then will last for years. The time required for a complete mummification of body varies from 3 months to a year and is influenced by the size of the body as well as the atmospheric conditions and the place of disposal of the body. The factors necessary for production of mummification are the absence of moisture in the air and continuous action of dry or warm air. The medico-legal importance of mummification lies in the fact as the features are well preserved, the deceased can be identified. The time since death, of course, is difficult to be calculated for the same reason. The cause of death can be determined because the injuries are well preserved. To summarize this topic, the rigor mortis appears in the body starting from top and going downwards. It is fully developed in about 12 hours in whole body, persists for next 12 hours and afterward disappears from whole body in next 12 hours following the same sequence of appearance as well as disappearance. The decomposition starts around 24 hours after death, comprising of the putrefactive and autolytic changes. The marbling, collicutive liquefaction and ultimately skeletonization occurs after death up to 1 month and then goes for years. Then depending upon environmental factors, instead of decomposition, the dead body may go for Adipocere formation as well as mummification. The cadaveric rigidity is one of the conditions which mimic rigor mortis and hence needs to be specifically differentiated.